So today we are having one topic which is related to again the giant nerve fibers in the case of the decapods. So here we'll be taking the example of the crayfish to demonstrate that how the giant nerve fibers they are helping in the reflex actions, or you can say the predatory response of the crayfishes. So students, the crayfish, these are actually the decapod crustaceans that are using different forms of their escape mechanisms to escape or flee from the different types of the predatory attacks. So actually, whenever a prey it attacks the crayfish, it can give different types of escape responses. So those escape responses, they should be in a particular given time, and they should take very less time to happen so as to save that particular crayfish. So the lateral and the median giant fibers, actually they are helping the crayfish for fleeing out of this particular action. So students, first of all, I think we should start with the basic structure of the lateral giants or the total giant fiber system in the crayfish. So first of all, if you go by the first diagram related to the crayfish, so here you can very well see, if you go by the A diagram into account, if you take this diagram into account, you can see the position of the exact the brain. Then you have the thoracic ganglia, we have 5 thoracic ganglia, then we have got the abdominal ganglia on the lower side. Now if you take the case of the figure B, now this is a cross section of the cord, you can see. So here you have 4 giant fibers in the nerve cord of the crayfish. Now out of these 4, 2 are called as the median giant fibers, so which are on the top in the mid section of the nerve cord. So these are the median giants, and 2 are called as the lateral giant fibers. So these are also written, you may find a word which is written in the books which is called as LG. So actually these are the lateral giants, so they refer to as the lateral giant fibers. So in total we have 4 giant fibers in the case of the crayfish, which are responsible for the fleeing actions of the crayfish in a predatory action. Now in the C part of this particular figure, you can very well see that these are some of the electrophysiochemical experiments going on the giant motor systems in the case of the crayfish. So they have taken one particular ganglion, which is shown on the below right corner. So this is a single ganglion. This is I think this is abdominal ganglion of the crayfish, and it has been sectioned. It has got 3 roots which are coming out on the left side, and 3 roots are on the right side. They have not been shown over here. So you have the root 1, 2 and the 3rd root. So 3rd root has been labeled over here in the diagram. So every abdominal ganglion has got 3 roots on the left side and 3 roots on the right side. So this particular diagram is giving you the overall idea of the giant nerve fibers in the case of the crayfish. Now after this we will be going to the second part of this diagram. This particular picture is giving you a brief idea about the structure of the abdominal ganglion of the crayfish. Now actually this is a single ganglion. This is a big ball kind of a structure, and you may find 4 giant fibers on the base of this particular ball-like structure. So these are the 4 giant nerve fibers which are going through these ganglia up to the end of the body. So here there is one giant motor neuron. So the white structure which is prevalent over here with some like branches, so this is the giant motor neuron, and it is also showing the relation of this giant motor neuron with one of the 4 giant fibers which are present over here. So you may find the connection in between this giant motor neuron and the lateral giant fiber. So the lateral giant fiber on the left side, out of the 4, it has got a connection with the giant motor neuron. So this motor neuron has been found to like elicit some of the impulses in the case of the fleeing action of the crayfish. So I'll be taking that particular part later on in the lecture. So first of all, I must give you the structure of this particular abdominal ganglion in full, and then we will discuss that how the impulses have been generated and how they are recorded, and what is the mechanism behind the recording of the impulses. So now if you take the figure 3 into account, here students, you can see a particular ganglion in the very center. So this is the 3rd ganglion taken. So this is the 3rd abdominal ganglion. This is a single ganglion, and here you may find the 3 roots which are coming out on the left and the right side. So here they are labeled as R1, R2 and R3. So these are the 3 roots of the ganglion, and in the very center of this diagram you can see 4 parallel giant fibers running through the whole of the nerve cord. So these are the 4 giant fibers, and out of these 4, 2 are in the center which are called as the median giant fibers, and 2 are the lateral giants which are also known as the LGs, and median giants are known as the MGs. So students, this is the overall structure, and again if you see the right side of this diagram, on the very right, this is the next 4th ganglion of the abdomen. So we have a 3rd ganglion in the center, you have the 4th ganglion which has not completely been shown in this particular diagram, but you can see the 3rd root of the 4th ganglion. Here it has been written as the 2nd, so actually we are going towards the head on the right side. So here we have the 2nd ganglion on the right side, we have the 3rd in the center, and then the 4th ganglion will be on the left side of the diagram. Okay, now students, after this basic information about this diagram, now here we have got 3 major motor neurons which are in the connection with the giant fibers. So these are labeled as the F3 motor neuron, F5 motor neuron and F9 motor neuron. So these are the 3 black colored motor neurons which are present in connection with the giant fibers. Now, if I take the first case, now on the very left side there is a micro electrode which has been inserted into the lateral giant fiber. So you can see it over the left side. So here it has been written as number 1, Roman number 1. So this is the first stimulus that has been given to the giant fiber, which is lateral in position, and this stimulation, it is resulting into a orthodromic kind of a response, both in the case of the motor neuron F3 and the root 3, which is going to the flexor muscle. So if you see the black box, which is having 2 graphs on the left side, upper left side, so you can well see the orthodromic shift in the case of the soma and then the root. Soma refers to the shift or the voltages which are present inside the F3 motor neuron. So that means if you give an impulse at level 1, then there will be a voltage produced in the F3 soma, and the second voltage is present in the root of the flexor muscle, or the root of the third ganglion, you can say. So that is the orthodromic shift in the case of the first impulse response. Now, if you take the second case, it is on the third ganglion itself. So here the second stimulus is given at the level of the F5 motor neuron. So this is directly given into the body of, or the soma of the motor neuron F5. Now, what is the response actually? So when you give stimulus or the voltage to the F5 motor neuron in the third ganglion of the crayfish, the response can be seen both ways. First response is in the soma itself, in the F5 motor neuron. So here you can see a black box which is on the lower side. So the top graph in this box is for the soma of the F5 motor neuron, and if you see the lower graph, it is for the muscle of that particular side, the flexor muscle of the left side, through the root 3. So this is a big graph, and you may expect a muscle contraction in this particular case. Now students, we are taking a third stimulus at the level of the root axon or the motor giant fiber in which is shown on the right side. Now if you take the third stimulus, where is it is shown on the right side of this particular diagram. So this is the third stimulus. It is given into the root axon, and now from here the impulses are traveling back into the nerve fiber, and then they will reach the F9 giant motor neuron. Now what are the responses in this particular case? The first response is the antidromic shift in the case of the soma of the F9. So here is a you can say a low voltage which has been developed in the soma of F9, and the muscle is also activated through the stimulus. So actually the stimulus was given into the root axon, so it will travel to the muscle or the flexor muscle which is present over it. So the third black box, it is showing the antidromic shifts in the case of the third stimuli given to the root axon of this particular system. So students, this particular diagram is giving you the 3 cases of the stimuli given at different points. First was given at the giant fiber level, second was given to the neural soma of F5, and third is given to the root axon, and all the 3 stimuli, they are showing you orthodromic, direct shift, and the antidromic kind of responses. So this particular diagram is just an electrophysiochemical experiment which has been done on the crayfish to show the electro responses of different motor giant fibers, the motor neurons, the flexor muscles. Ultimately, what is the importance of this system, or the importance of the electrophysiochemical experiment which has been done over here, is that whenever the crayfish is in some danger, through these impulses through these giant motor systems, through these giant motor fibers, the crayfish can flee out of that particular place, and it can save itself from the predator. So that is a kind of reflex action that has been shown by the crayfish, and this is under the control of the giant fibers. So that was the case of the flee action. And now students, if you go by the fourth diagram, so it is showing you the crayfish escape response circuit. Actually, this is showing you the connections in between the different parts of this response circuit. First of all, we have the tactile receptor, which will receive the stimulus. It can be a mechanical pricking, it can be some mechanical touch by the predator. So after sensing the particular stimulus, the tactile receptor will give off the impulses to either the tactile interneurons. Here they have been labeled as the A, B and C. They may interconnect in between themselves. So the impulse is now in the interneurons, or the tactile receptor can give a direct impulse to the lateral giant fiber. So here it has been written as LG. So there can be like different types of motor neurons for the impulse traveling. So the alpha motor neurons are giving a direct connection in between the TR and the LG, whereas the beta motor neurons are present in between the A, B and C interneurons and the LG. So it has been written in the diagram itself. So the lateral giant fiber, or the LG, is giving stimulus to two different moieties. Now the first moiety is the giant motor neuron itself. So it can activate the giant motor neuron, and that motor neuron can activate the muscles of the body to flex the body parts and flee away from the place. Otherwise, the LG can also stimulate the FFMN section of the motor neurons. So what is FFMN? It is the fast flexor motor neuron system. So this is the second part, which is in parallel to the giant motor neurons, and this fast flexor motor neuron can activate the muscle itself for the flee action. So either you can say the crayfish will flee out of that particular place to escape from the predator, either through the LG mode, or it can go through the MG mode, which is the median giant fiber system. So ultimately the muscles will contract to their full in very less response time to take away the crayfish from that particular place, and that will flee away, and that will go out of the reach of that particular predator. So that was the tactile escape response circuit for the crayfish, and that is altogether controlled by the giant fiber system. So students, that was all about the crayfish.